I think we can probably begin. We have everybody here present. So, hello, good afternoon, everybody. I'm Sophia Foch, the Sales Associate or Cryo-Free Crusader, according to my email signature here at Atelerix. And I'll be hosting this webinar this afternoon, along with our presenter and product specialist, Dr. Sarah Marsh. Our webinar title for today is Enabling Global Vaccine Access, Hypothermic Stability of LNP-mRNA and Viral Vectors with CytoStor. The presentation will be around 20 minutes, followed by a Q&A session. So feel free to drop any questions you have into the Q&A box below, and we can get those answered for you after the presentation. So without any further ado, over to you, Sarah.
Thank you, Sophia, and hi everyone. Thank you for joining our webinar today. I'll share my screen and then we'll go through the presentation. There we go. So thank you everyone for joining. Our webinar today is focused on preserving LNP-mRNA and viral vectors with our product CytoStor. And ultimately, this is helping to enable global vaccine access using hypothermic stabilizing technology. So for anyone that's new to Atelerix, we are a biotechnology company based in the northeast of England. And we have developed technology to preserve a range of different biospecimens at hypothermic temperature. So ultimately, we're here to turn the cryologistics world on its head and become every lab's favorite shipping companion across the globe. So we're really trying to define how labs operate, all while trimming the costs and also improving the outcome on the environment by reducing carbon emissions worldwide. So how does it work? Well, at 37 degrees at normal physiological body temperature, cells have an intact lipid membrane and they are functioning as normal and are metabolically active. However, as this temperature decreases below 32 degrees to, for example, 20 degrees, which is the average room temperature, the cells enter a state of hibernation, but at the same time, they become susceptible to damage to the cell membrane as there is dysregulated transport of ions and water in and out of the cell, and that ultimately leads to cell death. So the way our gels work, they are an alginate-based hydrogel that help to stabilize the cell membrane so that when the temperature does decrease below physiological level, you don't get this membrane damage and subsequent death that would usually occur at this temperature without the protection. So because our technology enables this protection to the lipid membrane, we've been able to apply it to a range of different biospecimens, including primary cells, cell lines, tissues, viruses, organoids, also blood products, for up to 2 weeks without the need to freeze or fix. And the latest application that we have applied this technology to is viral vectors, viral particles, and viruses. So what are the challenges associated with currently storing and distributing vectors and vaccines? Well, because these because the virus and vaccines aren't stable at room temperature, it requires cold chain logistics. And this is associated with a wide range of disadvantages. So, for example, very expensive cost associated with shipping viruses at ultra cold temperatures because it relies on dry ice and ultra cold freezers. This temperature control is also very energy intensive and carbon heavy and a real detriment to the environment. So, for example, 99% of the total carbon footprint of the COVID-19 vaccine distribution was associated with distribution. In addition to this, the cold chain logistics process is very complex, and any sort of logistical failures or breaks in the cold chain at any point can lead to degradation of the samples and therefore wastage. So it's estimated that around 50% of global vaccines are wasted. And finally, it's very hard to deliver and store vaccines in areas without the infrastructure in place. So poor electricity or no cold storage. So often these rural areas lack the ability to access these vaccines, leading to unequal healthcare. So our solution is able to overcome these disadvantages by enabling storage and distribution at hypothermic temperatures, which of course doesn't rely on these ultra cold logistics. So we started with firstly studying encapsulation of coronavirus, which later led on to us looking at lentivirus and LNP-mRNA. But firstly, just to show, the CoV-2 virus is very stable in CytoStor, and encapsulation enables the storage of this virus for 2 weeks. And this is a 5-fold extension from the PBS negative control. Alongside this, we have also shown considerable protection of host cells of the virus, so human airway epithelial cells. And again, we see protection in CytoStor for up to 2 weeks compared to the PBS control. We're then looking at, oops, sorry, just go back. On the graph on the far right, looking at CoV-2 infected host cells after transduction of the virus into the human airway epithelial cells, we showed that the cytopathic effect of the virus is retained after 2 weeks, which is a dramatic difference from the buffer control, viral transport medium, whereby the cytopathic effect rapidly deteriorates within the first 3 days, actually to below the limits of detection. So after these observations, we applied the technology to lentivirus and lentiviral vectors. These are a very popular tool for delivering genetic material into host cells and are being increasingly used in gene therapy and vaccines. So as we can see here in CytoStor, there is no loss in the levels of viable lentivirus after 14 days when it's stored in CytoStor. And this is compared to the PBS control, whereby the titer of virus rapidly declines within 3 days below the levels of detection. Looking at lentiviruses too, we can see that they can be preserved for up to 14 days in CytoStor 2 to 8 degrees, which is a considerable improvement from the negative control. So the transduction efficiency after 1 week has dropped by sort of 60% in the control. And even by day 14, the transduction efficiency is retained and there's no decline by 2 weeks. More recently, we have applied our CytoStor technology to LNP-mRNA preservation, which is a very useful platform to deliver genetic material, mRNA, into host cells and stimulate immunity to a virus, such as the COVID vaccine. It's a good example of this. So In standard buffer alone, after 1 week at 2 to 8 degrees, there is a considerable drop-off in the percentage of transcript positive cells by around 50%. However, using CytoStor to protect the LNP-mRNA, there is no decline in the transduction efficiency, and the LNP-mRNA is successfully stabilized, and that continues up to day 30. In addition to this, we have also shown that this process is compatible with long-term storage. So the graph on the right shows initial freeze thaw of the LNP-mRNA, followed by storage for up to 2 weeks, and then freezing and thawing again. And we can see that the transduction efficiency of the LNP-mRNA is still retained even after integration with long-term freezing. So this means that, for example, you could generate large amounts of LNP-mRNA stock, store at -80, and then rather than having to ship those samples on dry ice, you could simply encapsulate the samples in CytoStor and ship at 2 to 8 degrees, and ship for up to 14 days. So it's sort of like a pick and ship option, which is much cheaper, much more flexible, and better for the environment too. So how does the protocol for CytoStor actually work? Well, it's very straightforward. To begin, you will receive your kits, which contain 3 components, those being gel-based beads, gel A, and a release buffer. So you would start by taking an aliquot of a purified virus for your sample, typically within sort of the 1 ml range, and you would add the virus to the gel A and mix. You would then add that solution to the gel-based beads, and that will initiate gelation. And this process only sort of requires around a couple of minutes hands-on time. It's very straightforward. You will then store or ship the sample and send it to the end user. And at the end user site, you would just add a release buffer, which will dissolve the gel within around 10 minutes, so that it goes back to a liquid form. You will then retrieve the virus sample, and it's ready for downstream application. So just to quickly summarize, we're able to stabilize viruses for up to and over 2 weeks, extending shelf life by 5-fold compared to buffer alone. We can preserve lentivirus and lentiviral vectors for 2 weeks, and LNP-mRNA is stabilized for up to and over 30 days. Our CytoStor product enables a pick and ship option without dry ice to ship LNP-mRNA at 2 to 8 degrees. And this really opens up and enables cryo-free virus and viral vector distribution, improving the flexibility of your workflow, making it more cost effective and better for the environment. In addition to this, our technology is fully scalable via liquid handler automation. So we're able to supply as customers require. And we operate through a volume-based discount to suit customer requirements. So the price of the kits will be sort of tailored to the volume that's required for your process. So just to summarize, you are able to store, ship any sample anywhere without any freezers, dry ice top-ups during transit, really helping to alleviate any stress associated with shipping samples. Overall, we are 10 times cheaper. We lower the cost of shipping by removing the need for dry ice and ultra cold freezers. Again, increasing flexibility. So there is an increased buffer time at distribution sites, routes, and customs. It doesn't require personnel to be at a certain time, at a certain place exactly, because there's flexibility there using our CytoStor. Again, our products are scale ready and easily automatable with the liquid handling. Volume-based pricing is available, and ultimately you can get a free sample yourself and see how well it works for you. We have a number of resources available if you want to find out a bit more information. We have our data pack with the data I described in. The protocol is available if you'd like to take a look at the steps involved. And we have a white paper which will be released in the next few weeks. And if you are ready to give it a go for yourself, please get in contact with Sophia or myself, or you can contact sales@atelerix.co.uk, and one of our members of our team will be in touch with any queries that you might have about today's webinar. So thank you everyone for listening. We will now go through the questions.
Thank you, Sarah, for that fantastic and informative presentation. So on to the Q&A and for Sarah to answer some of your submitted questions. And do feel free to continue sending them in and we can get them answered in this time. So the first question is there any special equipment required for the protocol? Sarah, you're just on mute.
Oh, thank you for the question. No, so there's no special equipment required. It's just the standard pipettes that you find in a laboratory just to mix the gel in your solution. There's nothing extra that you need for the protocol.
Okay. The next one is, are there any known safety risks or toxicity associated with hydrogel components post-encapsulation?
Another good question. So all of the components of our kits are made under GMP conditions, and all components are biologically inert. So the kits pose no safety or toxicity risks.
Thank you. Have you looked at LNP-mRNA storage in CytoStor at room temperature as well as refrigerated?
Yes, so for the LNP mRNA preservation, we have looked at room temperature, 15 to 25 degrees as well. We do see that 2 to 8 degrees performs slightly better, but you are able to store at room temperature, and it does have the additional benefit of applying that to areas without 2 to 8 degrees storage in place. We just see optimal performance at 2 to 8 degrees.
Great, thank you. What is the shelf life of these kits?
So the shelf life is for CytoStor is 6 months.
Thank you. And do you offer a free trial I can test first?
Yes, absolutely. So anybody that wants to get a free sample of CytoStor, please get in touch. We can organise sending that across. Very happy to provide that.
Great, thank you, Sarah. if we, not sure if we have any more. Oh, we have one more question just come in here.
Mm.
Can you please clarify your point about scalability with liquid handlers?
Thank you for the question. So we have compared the production of producing CytoStor manually versus our liquid handler, and it manufactures our products to the same standard. So we're able to scale up that process of production to whatever level is required. If you want, we can email over any specifics if there's sort of a certain requirement that you have.
Great. Thank you very much for that. Okay, so if we don't have any more questions for now, then we can draw that part of the webinar to a close. If you think of any questions after the presentation, then please either contact us through our website or sales@atelerix.co.uk, and a member of our team will get back to you on that. So just to close, thank you, Sarah, very much for your time. And we have actually recorded this presentation, so it will be available on our YouTube and socials if you'd like to recap anything we've discussed here. And if you are keen to know more or looking to trial our products, we can organise a free sample for initial testing and for you to give our hydrogel encapsulation method a real go. So thank you everybody so much for your time and for joining the call, and we'll catch you on the next one.
Thank you, everyone.
Thank you, everybody.